Hi everybody, we're back here in the 224 lab for what has affectionately become known as the blood lab. Because yes, I am going to bleed for you. So join me if you would on page 1 of lab number 4 in your trusty lab manual and realize that today's lab is both blood and the lymphatic system. So we will look at a few physiological aspects of blood, anatomical models, a few of them for the lymphatic system, and histology for both. The identification of blood cells, as well as the identification of lymph node, lymphoid nodule, spleen, like that. So, before we begin, it might behoove us, and yes, I did just use the word behoove, for me to go over a little bit of the physiology behind the principles in this lab before we start the procedures. Now, I do not want to steal the thunder of your lecture instructors because, as you have probably figured out by now in the semester, we lecture instructors really love to hear ourselves talk, even if it's online. But depending on where you are in your lecture, you may or may not have some of this information. So if you already know all this, bear with me, or in an online format, why not just fast forward me? But what you can see here are a couple views of red blood cells, or erythrocytes. Whether you want this top view or the classic side view biconcave disc. You probably already got that in your lecture. Now, these are cells, not exactly like all of your cells because they have no nucleus, essentially a bag of hemoglobin, etc., etc., lecture stuff there. But because they are cells of yours or mine or the next person, they have oftentimes, in fact most all times, proteins on their cell membrane that identify these cells as to what they are. Those proteins that live on the surface of your cells and my cells and other cells and pathogens, etc., these are called antigens. Proteins that live on the surface of the cell, identifying it for what it is. When we're talking about antigens on erythrocytes that cause this clumping or agglutination of blood, those are specifically known as agglutinogens. They generate or cause agglutination. So the vocabulary word that's boldface in your lab manual, agglutinogens, these are the antigens found on the surface of red blood cells. And depending on what your blood type is, you may have one or more or none of these antigens present. So when we talk about typing blood, we talk about the proteins that are on the surface of a person's red blood cells. We are looking at the classification of 3 specific agglutinogens or antigens that might be present. The A, the B, and the RH antigens. Simply put, and you can see I used high-tech drawing here, a curly Q for the A antigen, a lightning bolty zigzaggy line for the B antigen, and just a general brown curvy thing for the RH antigen. So let's say I had a person's red blood cell right before me, and on the surface of their red blood cell, so a protein on the surface of the red blood cell, let's say they had this one right here. This blood cell is type A blood because it possesses the A antigen. If I erase these, and again, sorry if you already know this, let's say that this person has on the surface of their red blood cells these specific zigzaggy proteins, the B antigen, this person has type B blood. So all we do is identify what protein or proteins a person has on the surface of their erythrocyte, and that's the blood type they have. Let's say that this person has the B protein, the B antigen or agglutinogen, and they have the A. So the surface of their red blood cell has both B and A antigens, then this person has type AB blood. They have both. But let's say that this person is like me. They don't have either one of them. So give me a moment to erase all these. So let's say this is the red blood cell. Let me grab a red one, because I erased part of my cell membrane here, people. Let me put it back. Let's say this is their red blood cell. This is mine. Notice no A, no B antigen or agglutinogen on the surface. What blood type is this? O. This is blood type O. And O, don't think of it as a letter here, think of it as a number. They have 0 of the antigens. No A, no B, blood type O. That's me. Now, I know a lot of you know this because you were at our, virtual club meeting last week for type O blood, O power. Yes, that's our secret signal. But if you have neither one, you're blood type O. Now, you'll notice I didn't mention this Rh protein. This wasn't discovered till much later. Some people were found to have it, and some people were found to not have it. So, this Rh protein, Rh named for the rhesus monkey, not the peanut butter cup, spelled differently. But let's say that my person has this. The only thing on the surface of his or her red blood cells is this Rh antigen or agglutinogen. Then we say that this person is positive. The positive or negative you hear in blood types refers to this antigen right there. We describe a person as positive if it's on their cells. We describe a person as negative if it's not on their cells. So this person has no A, no B, so their type is O, and their positive or negative is positive because they have it. This would be an exact picture of my red blood cells because I am type O positive. No A antigen, no B antigen, but I do possess the Rh antigen. What if this was a person? Of the 3 antigens we're talking about, they have none at all. So their type is O, and they are negative. O negative is this one. Just to reinforce it, let's try this, everybody. How about this blood type right here? All right, so see this person? They possess the B antigen. Their blood type is B. They also possess the Rh antigens, so they are B positive. Everybody get it? How about this? B negative. That's how it works. Now, how is it that we're going to test some blood cells to see what type they are? Well, for that, we will use antibodies. Proteins that a person makes, their B lymphocytes primarily make, that will bind to foreign antigens. I'm going to repeat myself. Foreign antigens. When we're talking about antibodies for blood typing, they're called agglutinins. So notice the similarity between these two terms. Be careful on your lab quiz. Agglutinogens are the antigens on the surface of the red blood cell. Agglutinins are the antibodies that we will make against antigens that we do not already have. When a person gets vaccinated, let's say for coronavirus, the goal is to stimulate them to produce antibodies against the virus. Not antibodies against what we have, antibodies against what we don't. Now again, I really don't want to steal the thunder of your lecture instructors, but agglutinins are proteins, antibodies that will bind to these antigens, or agglutinogens, throwing the vocabulary around. So if I put anti-B antibodies near these cells, they will bind to these proteins and cause this clumping of the red blood cells that we look for. If a person is this person, type B negative blood, do they make antibodies against B, which they already have? No, not unless they have a disorder, an autoimmune disorder. What would they make? They would make antibodies against A antigens because they don't have them. They would make antibodies against Rh antigens once they're sensitized because they don't have them. They will not make antibodies against B. Now let me try to explain a little bit of how this will work in a practical sense. Okay, so look at my very artistically rendered lab setup. I can't draw that many things well. In fact, some of you people may already be saying, Dwayne, you can't draw anything well, but I can draw a reasonably good finger. So look at that thing. So here is the gaping wound that I'm going to try to make in my own finger in just a few minutes for you here in lab, out of which I want to get 4 drops of blood. Drop number 1 is going to go into the hematocrit tube that we will use to determine my hematocrit, the percentage of my blood that is red blood cells. I'll explain a little bit more about that when we read the hematocrit. But the first drop, I want to go right into this capillary tube, this hematocrit tube, and it gets drawn in by what we call capillary action. Thank you to the adhesion and cohesion of water molecules, to be a little science nerdy here. The other 3 drops, 1, 2, 3, I want to deposit in my blood typing tray. So I'm going to have a typing tray with 3 wells or depressions or fossas, if you will, in this thing that I have preloaded with antibodies or agglutinins for blood typing. So in the A well, I place the anti-A antibodies. In the B well, the anti-B antibodies. And in the RH well, sometimes referred to as D, letter D, I put the anti-RH antibodies. Then I will introduce my blood drop, drop, drop like so. And of course, in my blood droplet, there will be a whole bunch of red blood cells, and on their surface, they will have or will not have specific antigens or agglutinogens that may or may not react with the antibodies. So I preload anti-A antibodies. So if I put red blood cells into this well that contain A antigens, then the anti-A antibodies and the A antigens will bind to each other, causing this clumping appearance, which is one of the things antibodies might do, that agglutination. So again, let me say it, I'll just use this well. If my red blood cells possessed the B antigen, then the anti-B antibodies would bind to them, causing the clumping. Same thing is true over here for the Rh antigen. So if I preload each well with the agglutinins, the antibodies, if I then introduce the antigen or the agglutinogen, I will get a reaction. Now, I already told you people, my blood type is O positive. So do I have the A antigen? I'll wait. No, I don't. Do I have the B antigen? No, I do not. But I did say I was O positive. So do I have the Rh antigen? Yes, I do. So what I expect to see is no clumping in this well, no clumping in that well, but I do expect to see clumping in this one, because I already know that I have no A, nor do I have B antigens on the surface of my red blood cells. If this doesn't make a lot of sense to you, rewind, play me over, make sure you read the section on page 1 of lab number 4. So we are trying to get an antibody antigen or agglutinogen reaction to happen in these wells by preloading antibodies and then dropping in blood. Let's hope it works. Okay, here I am all ready and set up to do some bleeding for you. So we, your instructors, not only do we sweat and toil, I'm ready to bleed. And my target is the medial side at the distal end of my left hand ring finger. Why do I choose this site? Well, I happen to be right-handed, so I don't use my left hand that much. My ring finger's side is much less likely to be irritated throughout the day, so that's usually a clinical site of choice for this stabbing procedure. What am I going to stab myself with? This device right here, sometimes called a lancet or an autolet, much better now than the ones I used way back when in the old days. When I originally had to stab myself when I was an undergrad, we had what amounted to be a pointy piece of sheet metal, and you had to stab yourself with it, and man, did we get a lot of blood. These are much smaller, much more sterile, much easier to use. In fact, I'll hold it sort of by the microphone. Listen very closely. Maybe you heard a little click there. So when you press this against your finger, a little blade shoots out, stabs you in the finger. Now remember, what I just told you is I want to get 4 drops of blood out of this finger right here. The first one will go into this capillary tube. So I'll put this capillary tube right up against the drop of blood, and because of the nature of water, which plasma is mostly, hopefully a good column will be drawn into this glass tube, which I will then seal with my crit-o-seal clay. I don't know if you can see it. You will when I actually do it, because I will change the camera angle. But I've sealed one end of this capillary tube with blood in it in a few minutes. Then I want 3 more drops into the 3 wells on my typing tray. A drop in the A well, drop in the B well, drop in the RH well, which I will have preloaded with my anti-A antibodies, blue, anti-B antibodies, yellow, and no, they don't naturally occur in these colors. These have been colored so it's easy for a person to tell them apart. And the anti-RH antibodies, clear. So I put a few drops, 2 or 3, in each well. A antibodies in the A well, B antibodies in the B well, RH antibodies in the RH well. Then I add my drops of blood. I have 2 sterile toothpicks right here that I will use to stir up the blood. So I'll stir up the blood in the antibodies, turn my toothpick around, stir up the blood in the antibodies in the next well, then grab a new toothpick, stir up the blood in the antibodies there. Why do I use 2 toothpicks? Well, I can't allow cross-contamination between my wells, or that might mess up my results. These antibodies I just pulled out of the refrigerator back there. They must be kept cold because they're proteins. And if we allowed them to reach room temperature and get too warm, they might denature, come apart, and then no longer function, because the functioning of proteins, as you know, is always tied to their 3-dimensional structure. So 4 drops of blood, 1 in the capillary tube, 1 in each well. Then I need to seal up my gaping wound with a Band-Aid, put gloves on my hands. Now, before I do any of this, of course, I need to prep this finger. I'm going to clean it with an alcohol wipe. Try to get some blood in there. Try to warm it up, and then stab myself 4 drops, 1, 2, 3, 4, put the Band-Aid on. Then I can proceed to try to read the typing tray and with the hematocrit determination. So I'll change the camera angle so you can actually watch this happen. Okay, so hopefully you can see my setup here ready to go. Got a capillary tube right there, a couple toothpicks, my blood anti-sera or antibodies preloaded into my typing tray. Got my finger all ready to go. And here, of course, is my weapon that I used to stab myself to get some blood started here. So you see the blood drawn up into the capillary tube. Want to get a good amount in there if I can. A tall enough column to use a good hematocrit chart. So sometimes you have to squeeze your finger a little bit. Okay, that's probably a pretty decent amount of blood that will work. So I will seal it with my centrifuge clay, my CritoSeal. Set that aside for now on a sterile. And then get my blood drops into the typing well. If they don't want to fall off your finger, you can just draw the drop down with your toothpick, like I just did right there. There's one. And 2. Let's get a little more in there. Set this aside. This is now contaminated. A biohazard, and 1 last drop is what I need Let's see if I can get one, a good one, into that RH well. Keep working. If you start clotting quickly, like I am here, it's sometimes hard to get your last drop off. But there we go, good amount of blood in that well. Stay tuned for what happens next after I put on my Band-Aid. Okay, this device that you see right here is a centrifuge. You can see I put on my other glove after I bandaged up that great big wound. And this device, let me do some fancy camera work here. You can see has a bunch of slots in it into which capillary tubes go. So I have my capillary tube right here that I will place into slot number 1. And then I have a dummy capillary tube that I just put some water in that I will put across from it so as to keep it balanced. I will seal this, and then I will start it spinning, which will separate the components of the blood, the more dense cells being pulled to the outside of this circle, the less dense plasma going to the inside. I have to let it run a few minutes. So while we do that, let's walk ourselves over and check out my typing. So here you see my typing tray. I've placed it on what we call a rocker box or a light box. It provides light and a heat source. Let's zoom out so you can see it. One of those that makes it easier for us to read this blood typing tray. Notice in the A well right here, there's no clumping of blood, no graininess. And the same is true in the B well. No clumping. But look at that RH well. I'm going to zoom in as close as I can get. There it is right there. Notice how the blood in there has clumped. It has become grainy looking in appearance. This is very characteristic of an RH reaction. Sometimes, particularly in A or B, you might see the blood just meld up and merge into one giant clump. But since I see no clumping in the A, I do not have the A antigen. No clumping in the B, no B antigen. So my blood type is O. Since I do see that grainy appearance, look closely at it, in the RH well, I am RH positive. So my blood type is O positive. And I'll show you a few other examples here in the video to make it a little more clear for you. So if I saw clumping in A, blood type A. If I saw clumping in B, blood type B. If I saw it in both, AB. Since I see it in neither, O. O positive. Okay, so I want you to notice my hematocrit tube fresh out of the centrifuge. Looks just fine, and in fact, it did not explode, as they sometimes do. What I want you to notice, and I will zoom in a little bit for you, hopefully you can see the much clearer plasma above and the packed red blood cells below. Make sure that your lecture professor tells you about what about those white blood cells and platelets, but I'll leave that for them. Notice that my plasma has a little bit of a red hue to it. That's from hemoglobin that came out of my red blood cells, most likely when I squeezed that finger or milked it, I did some what we call hemolysis or hemolysis, some blowing up of those red blood cells. So I have plasma above and red blood cells below. And the thing I have to do is determine how much, what percentage of this total blood column is the red blood cells. That would be my hematocrit. One way to do it is using a ruler, but it is much easier to do it using what's called a hematocrit chart, which is what I'm going to attempt to show you right here. So a standard hematocrit chart like this one, you will see has 0% at the bottom, 100% at the top. A little gap right here, which is for that hematocrit clay. So let me line this up and I'll show you how it's read. So I lay my capillary tube right on top of this chart. Everybody see that? Then I manipulate it with my hand so it's vertical. And what I want is the bottom of my red blood cells to be right at the 0 line. Now, it actually is, because of the angle of the camera, it doesn't look like that to you. But I'll show it to you in a minute. And then the top of my plasma to be at the 100%. So I want everybody to look at this as I've lined it up. Red blood cells at 0, plasma right at 100, and the creators, the makers of this chart, provide me with some vertical lines so I can keep everything straight. So if I do my best to line it up like this, now I'll try to hold it still, then I read, give my best approximation, I'll put it back for you to see in a minute, of where my packed red blood cells stop using these numbers over here on the left. If it was a much taller one, I'd use the numbers on the right. Now, does everybody see how I'm just a little bit over 45%? If I was reading this one, I would say that my hematocrit is about 47. About 47 is my hematocrit today. I line up the hematocrit tube, bottom of the red blood cells at the 0, top of the plasma at the 100, and I simply read it. And I'm right at about 47, maybe 46. I'll give you one more view of it. Closer up if I can. Oh yeah, see, look at that great camera work. Hey, when you're the cameraman and the director, it gets a little tricky sometimes. So everybody see that? My hematocrit right about 47 is what I would say. Make sure you understand how this is done, and I'll throw 1 or 2 examples of it in the practice video so that you can practice it before we have a live quiz. We are now in the next section of lab number 4, which is the identification of leukocytes. And you can see in your handy dandy lab manual that we have leukocytes spelled with a K. You can spell it with a K. The newer spelling is not with a K, but with a C. At this point in history, either spelling is perfectly acceptable. But leukocyte with the C is a little more modern for those of you that like the 21st century. Those of you that like the 20th century, like myself, maybe you go with a little more of the K spelling, but either one's okay right now. And you're probably saying to yourself, does he really believe in that draw it first to know it philosophy? Well, obviously I do, especially with micrographs. So I'll do it here with the leukocytes, but this is something you should be doing on your own in online labs, hopefully getting some benefit from it. And it gives me an excuse, a reason to use all these fancy markers. So start with me on the far left of the whiteboard, and you'll see a column that I've dedicated to the neutrophil, the most common of your white blood cell types. This one, about 2/3 of the white blood cells that you will run across, happens to be one of the granulocytes. They end in phil, neutrophil, eosinophil, basophil, those white blood cells that have visible granules in their cytoplasm. So let's draw a neutrophil right here. You should be doing this, don't just watch him do it. So we're generally talking about a somewhat large cell here, a neutrophil, that has visible grains in its cytoplasm, though they are sometimes hard to see and sort of widely scattered. The key to neutrophil identification is the fact that these cells are what is termed in science speak polymorphonuclear. In English, that means they have a goofy nucleus. So you will see this many-lobed, many-parted nucleus, sort of something like this, or even more bizarre looking, that would be a neutrophil when you see one of those. So all these white blood cell types are fairly large, except for this second one, a lymphocyte. This would typically, in a normal blood smear, be the smallest of the leukocytes that you will see, about the size of an erythrocyte, a red blood cell. So a lymphocyte, one of the A granulocytes, no visible grains in the cytoplasm. In fact, little helpful hint for this one, hardly any visible cytoplasm. So a lymphocyte, a smaller cell, they tend to be very spherical, usually, and they're basically all nucleus. You might see in a good one a little hint of sort of pale blue cytoplasm on one side of the cell, but a very large nucleus, lymphocyte, no visible grains in the cytoplasm, and you notice roughly about 1/3 or better of the white blood cells that we see. So you can see that neutrophils and lymphocytes, this granulocyte, this agranulocyte, account for the vast majority of white blood cells that we will see when we actually look at a slide. The monocyte, eosinophil, and basophil are very small in their percentages, not that common, but you can find them. These two, the monocyte and the eosinophil, are fairly easy to identify. A monocyte because of its size. The monocyte is a very large cell, even compared to the others. It's a very large cell. It's an agranulocyte, no visible grains in the cytoplasm, and typically in a perfect one, you would see sort of a large C-shaped nucleus like that. these very large phagocytic cells. In fact, the macrophages we talk about are usually monocytes or monocytically derived cells. So very large, no visible grains in the cytoplasm, monocyte. In fact, in your lecture, listen when you talk about immunity, about the many jobs of the monocytes and their progeny, the cells that arise from monocytes. So easy to identify here when you find a good one. Eosinophils, also very easy to identify. This is one of the granulocytes, so it will have visible grains in the cytoplasm. Usually a nucleus with two lobes, sort of looks like a barbell, a dumbbell, if you will. But the thing that gives away an eosinophil is the granules that you see typically tend to be sort of an orange or a reddish color. So you'll see a rather decent amount of orange, maybe pinkish, maybe reddish. See, I get to trot the colors out, see here, granules. So a two-lobed or bilobed nucleus, orange or reddish granules, very characteristic of an eosinophil. The basophil, the last of the white blood cells, the rarest of the leukocytes, no question. Very rare, look at this, typically less than 1% of the leukocytes we see fall into this category, but very easy to identify when you see one. Because the basophil, also a granulocyte, in fact, I would say a supergranulocyte, because the basophil is so full of granules, and when you study nonspecific immunity, you can learn what those granules contain. Hint, hint for your lecture. But so full of granules that you typically can't even tell where the nucleus is. It's just a bag of marbles is what I always think it looks like. So a bag full of granules, no visible nucleus. The two you are most likely to confuse one with the other is the basophil and the lymphocyte. So two things to focus on. First, the lymphocyte's size, smaller cell. And second, you can tell where the nucleus is because it's pretty much all nucleus, but look for that edge, that hint of cytoplasm you will not see in a basophil. Just a big bag of marbles. Last but not least, over here I have the platelets, which aren't really even cells, they're just cell fragments, and you just happen to find them around. There's not a specific number to memorize here with this one for your lab quiz, but they're just cell fragments hanging around. Once I show you some of them, I don't think you'll have any trouble at all identifying platelets. Okay, what you're looking at here is hopefully a good camera angle. I was actually jumping around on the tables here just a minute ago trying to get this perfect shot for you. This is a micrograph of a standard blood smear. People often ask, hey, Dwayne, is there such a thing as a dumb question? Well, of course there is. You know, an example would be, oh, Dwayne, did I actually miss that one on the lab quiz? Sure you did. But not a dumb question that people have. So if you wonder this, don't think that there's anything strange about you. This is a perfectly normal question that people have. Why is it that these are called white blood cells when they're all purple or blue? These little things that I see here. Well, the reason they're called white blood cells is because typically, other than the eosinophil, they have very little color. These have been stained, microscopically stained. So we make a blood smear, smearing a blood sample between two sheets of glass, and then draw a stain through them. So these cells that were white, you know, imagine your favorite white shirt, you spill a little spaghetti sauce on it, it's going to leave a stain. These white blood cells tend to absorb stain, so they're more easily seen. So they only look this way after we have stained them. Almost impossible to identify individual white blood cells on this magnification in this giant sea of erythrocytes. So we obviously have to go up to high power to get a look at some of these, and then that always requires some adjustment of the light, some refocusing, and then we'll start panning around a little bit, looking for what could be good examples of white blood cell types. Now, I see two here, whole bunch of platelets around. I see two leukocytes right here, a small monocyte and a neutrophil, but I don't like those examples for you. I want better, more, let's say, quiz-worthy examples for you to look at. So here we have a very fine pair of neutrophils. Notice that these two cells are larger than the neighboring erythrocytes that you see. Also notice all these little cell fragments everywhere, which are the platelets. Not even whole cells, just cell fragments. But focus your attention on this neutrophil and this one right here. It's tough to see individual granules in the cytoplasm, as I told you before, but look at the nucleus that you see in these two leukocyte examples. Very strange, multi-lobed, polymorphonuclear, goofy nucleus. These two right here are neutrophils. Now I want you to focus on these two leukocytes that you see right here and here. To your right, an obvious polymorphonuclear neutrophil. To your left, notice the size, about equal to the red blood cells, and right here at the bottom at about 6 o'clock, you can see that hint of relatively clear cytoplasm. This one on your left is a lymphocyte. Perfectly good lymphocyte. And neighboring just above, let me move my slide around. Whoops. There is a decent, though not great, monocyte. Let's find some others. Here is another, I think, quiz-worthy lymphocyte right here. So notice again the size of the cell. Down here at the bottom, that's just a coincidence, it's not always at the bottom, little hint of pale blue cytoplasm. Now, just to irritate you a little bit regarding your lectures, I can't tell looking at this lymphocyte if it's a T cell, a B cell, an NK cell, what it is, but I certainly can tell it's a lymphocyte, and again, some platelets neighboring. Here we have a very, very nice pair of lymphocytes, I think, just a different blood smear. You can see this one has a little more light passing through it. In fact, enough light so that the erythrocytes look like little donuts because their middle, remember biconcave disc, is so thin light can actually pass through it. So these two are very nice lymphocytes. See that little ring or crescent shape of cytoplasm there? And there happens to be a little platelet right next to it. So very, very nice lymphocytes right there. Okay, notice this cell that you see right here. I just happened to stop on this one because it definitely shows me the size. This one is a monocyte. I'm not a huge fan of this particular cell because it doesn't have the characteristic C-shaped nucleus that I would want to see for a quiz or a practical. But think about in your lectures, why is it that I, for one, as a lecture instructor, describe these cells as the incredible Hulk of immunity? Well, look at the size of these things. You can see how they could phagocytically go out and smash things. So very, very large monocyte, but I would want one with a C-shaped nucleus for your quiz or lab practical. Okay, look at this leukocyte right here. If you had to guess, if I forced you to guess as if it was a quiz, what would you name this one? Notice a two-lobed nucleus and definitely visible granules here. That makes it one of the phils, neutrophil, eosinophil, basophil. It's obviously not a neutrophil because I can see the granules in the cytoplasm. It's not a basophil because I can see the lobes of the nucleus. This one is an eosinophil. Characteristic, sort of reddish shaded granules that you see here, and a two-lobed nucleus. When you see one that looks sort of like this, you can see why I had a student in the past tell me this looked sort of like the head of a bug, an insect's head. And I can sort of see that if you think of the two big compound eyes inside the head, but that two-lobe nucleus, this one's an eosinophil. So look for that in the practice section, the colored granules that you see. So here's sort of a random pair of leukocytes that I came across. Eosinophil to your left. Notice this nucleus is two-lobed, looks almost like three, but the giveaway is those colored granules. And a lymphocyte on your right. Compare the size to the erythrocytes and see the little hint of cytoplasm. So lymphocyte, eosinophil. Now, I just had to stop on this one because it looks like a smiley face. Maybe just to cheer you up a little bit while you're stuck at home in an online lab. This one happens to be a neutrophil, goofy nucleus, obviously, and not really highly visible granules. So it is one of the granulocytes, but it's tough to see the granules in there. And I can see a platelet here, platelet there, platelet there. So this happy face, cheer up a little bit, everyone, neutrophil. Okay, here's one I think fairly to identify, easy to identify, excuse me. Eosinophil. Reddish granules, two-lobe nucleus. And this one appears to actually be leaking right now, which may or may not be the case. Tough to tell in this frozen moment in time. But if I had to guess, the compression of the two pieces of glass in making this preserved slide may have exploded part of the cell membrane right here. And this is a little bit of cytoplasm coming out. Eosinophil. So here's one for you that is very much the classic monocyte. Look at the size of the cell, this one large C-shaped nucleus. This is a monocyte. I think the picture perfect look right here, so you see a lot of cytoplasm, big cell, C-shaped nucleus. Here's just another very nice version of a monocyte. Large cell, bunch of cytoplasm, and you can see it if you look closely. C-shaped nucleus, platelets here and there. So another very, very nice monocyte. We had a happy face before. I guess I would have to say this one's sort of a grumpy emoji. This is another neutrophil. Notice the polymorphonuclear appearance. And right next door to it right here, another little platelet. So a neutrophil right there. So hopefully you can see this one right here, a monocyte. Take a look at it. And this one right here, I think you can see it, another neutrophil. Another very nice monocyte right here. See the C-shaped nucleus? Right here, and you have no idea, people, how much editing I had to do, how much I had to cut out to show you one of these. This is the always elusive basophil. So notice a couple things. First, the size of the cell, a little bigger than these red blood cells, and it's so full of granules, I can't actually tell for certain where the nucleus is. Less than 1% of your leukocytes, but here we have a basophil. For a quiz or a practical, I would prefer if it was stained a little bit darker. And the resolution that I will use for your practice section is hopefully better. But this one right here, that's a basophil. So I think we've seen each one of the types. The erythrocytes, of course, are obvious. Neutrophil, lymphocyte, basophil, eosinophil, and monocyte. If I just scroll around here, this is totally at random, not a plan. If I forced you, which in an online sense I am doing right at this moment, to identify this leukocyte that you see right here, what would you say? What is that one? That is a lymphocyte. Hopefully you got it and you saw the little ring of cytoplasm. Now again, I will include some practice for this lab number 4. So hopefully you will see them in the practice section and be able to tell one from the other. Okay, now I know what you're saying because I can hear you in an online lab, and you're saying, Dwayne, looking at leukocytes, identifying them as all perfectly fine and good, but let's get back to some good old straight anatomy, shall we? And I think we will. So join me here on page 4 of your lab manual under the lymphatic system anatomy. So here we have the model we affectionately call Mr. Lymph. I don't really get his pose with the hand up here, you know, this kind of, I don't get what he's doing here, but I think he's just trying to show you the armpit so you can see the axillary lymph nodes. So in this model, I see this whole network of vessels because your lymphatic system is your other vascular system. And this model doesn't come close to showing us all the lymphatic vessels, but it shows us some of them. So before I zoom in, let's take a look at some of these things that you're required to know for your next quiz. So this is very characteristic. You can see here all these lymphatic vessels, very similar to veins. You'll learn about that in your lecture. Typically in a model, they are color-coded either as white or yellow structures. So here, all these lymphatic vessels you see are white in color. So those are all lymphatic vessels. What you see as small yellow structures are lymph nodes. And remember, you have hundreds of those in your body. So oftentimes if we want to examine a particular lymph node, say to look for a virus, something like that, we just pluck one of them out of the armpit oftentimes, because guess what? You have, 400 to 600 of them depending, and you're not going to miss one. So these are lymph nodes, these little yellow structures you see here. And you will notice, I hope, that we have a lot of them here, 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 areas where the limbs or your head meet your torso. In the armpit, the axillary region, these are all called axillary lymph nodes. In the neck, they're called cervical lymph nodes. In the inguinal area, they are called inguinal lymph nodes. So axillary, cervical, and inguinal lymph nodes. And we also have another small grouping that live under the mandible. Those are submandibular lymph nodes. Cervical, axillary, inguinal are the big groups. We also have some riding under your mandible here called submandibular lymph nodes. And let's talk a little bit more about some specific structures here. So let's try a little camera work as I zoom in. So you will notice here in this model that a lot of the internal organs have been removed so that we can see this big lymphatic vessel running up the dorsal side of your body, sort of rides along with that azygous vein. This thing is called the thoracic duct. The thoracic duct is this large, the biggest lymphatic vessel of the body, large lymphatic vessel going straight up the dorsal side of your body. And, just for lecture purposes, I want you to notice that this thoracic duct makes a little U-turn right here, and it attaches right here near the subclavian brachiocephalic internal jugular junction right here. So this is the thoracic duct, biggest lymphatic vessel we have. Now, if my camera will allow, and it should, a little motion. So I'm going to move this thing down, and I want you to follow my probe down to the bottom right here of the thoracic duct. This structure you see right here is called the cisterna chyli. I'm going to say that again, then I'll explain a little bit. This structure, this bulge at the bottom of the thoracic duct is called the cisterna chyli. And not everybody even has one of those things. The cisterna chyli. So, as these lymphatic vessels are doing their job, which is returning lost fluid to the cardiovascular system, this fluid is coming in from the abdomen and the legs. And in some people, all these small lymphatic vessels merge into this sort of a little chamber or a sinus, if you will, a cistern, a collector of water. So down at the bottom of the thoracic duct, some of us, not all of us, have a structure called a cisterna chyli, a bulge at the bottom of the thoracic duct. The only other lymphatic vessel you have to know the name of is up here on the right side, and give me just a moment to do a little camera work for you here. So these lymphatic vessels, I don't want to turn this into a lecture because you all get plenty of online lecture, but you'll notice the thoracic duct connects to veins right here. And on the other side, I have this lymphatic vessel, notice this one right here, on my model's right side, called the right lymphatic duct, is the most common name you hear for that one. Some people say right lymphatic trunk, but right lymphatic duct, and the big one over here is the thoracic duct. You will see this in lecture. You have a very nice diagram of this in your textbook, but suffice it to say, as I zoom back out for you, it's great when you get to be your own cameraman, but the thoracic duct is responsible for collecting fluid, think about this in your lectures, that comes from about 3/4 of your body, the 2 legs, primarily the torso and the left arm. So this is much bigger. The right lymphatic duct is only collecting blood from the right arm and a little bit less than half of your head. So the thoracic duct is much bigger because it collects a lot more lymph, the fluid. The right lymphatic duct is much smaller because it only serves about 1/4 of a person's body. We're very asymmetrical here. I know some of you don't like that. You want perfect symmetry, but we do not have that in the lymphatic system. So what does Mr. Lymph show us? The nodes, the thoracic duct with the cisterna chyli, and the right lymphatic duct. So go ahead and rewind, pause me, play it back, all that sort of stuff. White lymphatic vessels, the thoracic duct, cisterna chyli at the bottom, right lymphatic duct right here, cervical nodes, submandibular, axillary nodes, inguinal nodes. Mr. Lymph. We also have what are considered to be lymphoid organs, your set of tonsils. And they're pretty easy to identify in my head and shoulders model here, the sagittal section of a person. So, depending on where you are in your lecture, you may or may not have talked about the nasal cavity and the pharynx and all of that stuff. We're just naming in the anatomy section of lab number 4 here, so notice right here where my probe is moving around, this is called your pharyngeal or pharyngeal tonsil, sometimes called the adenoids, at the back of your pharynx. So you will see with me on page 4 the pharyngeal tonsils right here. I always think that this tissue looks sort of like peach-colored scrambled eggs on most of these models. I don't know if that's because I want lunch right now or not, but pharyngeal tonsil. Notice your hard and soft palate right here, and at the bottom of those I have the palatine tonsil. Now, just a pet peeve of mine, do not confuse your palatine tonsil with the bottom of your soft palate called the uvula, that punching bag thing that you see at the back of your throat. That's not a tonsil. That's your uvula. This is your palatine tonsil right here. And third, but by no means least, right next to your tongue, you have the lingual tonsil. These are lymphoid organs. For lab, you just have to know their names when you see them. Pharyngeal tonsil, palatine tonsil, lingual tonsil. I will show you those on another model also. So here's another model showed me the same 3 tonsils. Pharyngeal tonsil or pharyngeal, you can say either one. Palatine tonsil, lingual tonsil. Pharyngeal, palatine, lingual. You shouldn't miss those. Now let's talk a little bit about your spleen, because when's the last time you talked about your spleen? So in the upper left quadrant of your abdomen, you have this spleen, also considered a lymphoid organ. This is the spleen. Notice, I don't know if you can see red and blue here, artery and vein. The red one, of course, is the splenic artery. The blue one, of course, splenic vein. I can even show you this one on another model as well. Okay, for the violent mobster movie fans, say hello to my little friend. This 3/4 size human is the one and only model that we have on this campus that shows us the thymus gland. I've got a lot of models in this room, but only one thymus gland. Remember that, because you hope I ask you this on your lab quiz or practical, because I only got the one view of it. So notice in my 3/4 human, the heart, and sitting right above it, this mass of tissue right here, is the thymus gland, another lymphoid organ. You will talk in lecture about what happens here and the importance of the thymosin hormones for T cells, but the thymus gland right here, and notice how big it is. This is way too big for a person of this size. This thing's greatly diminished usually in us adults. You can learn about that in your lecture, but he's got this giant thymus gland right here. So there's his thymus gland. Let me zoom in on it a little bit so you get a better view. So notice it right on top of the heart, the thymus gland. Right there, only one I have. So another very nice view here of a spleen. Notice upper left abdominal region, and I can zoom in on it for you. And you can see in blue the splenic vein, in red the splenic artery, the spleen. Okay, everyone, what you're looking at here is a perfectly good slide of a human lymph node. Join me on page 5 of your lab manual, right in the middle, lymph node. How can I tell that? Well, first, this particular organ is roughly bean-shaped, but it's very tough to tell that on a micrograph because you can only see part of it. And what you'll notice that I do in this histology section, and I will continue to do going forward in our labs, there'll be lots of cutting and splicing and changing in my video editing. And it's not just Dwayne trying to show off as a quote-unquote filmmaker. It is me saving you the problem, or the, how should I say, boredom of watching me change slides and focus and adjust the light and all that sort of stuff. But here I want you to notice a couple things we can see pretty easily on this slide, which isn't even that great of a lymph node, it's just okay. First, notice that the organ has a capsule, a fibrous capsule around the outside of it. Very common in many organs we will look at. And just like your brain back in 223, this organ has a cortex, the outside part, and a medulla, the middle part. The cortex is where you see these circular structures. Those are called germinal centers. And you will learn in your lectures what those are for, what goes on there, T and B cells hanging out, etc. But for us, in our lab, that's how you recognize a lymph node. It has circular structures called germinal centers in the cortex. So what could I ask you on a quiz or a practical? I could say, name this organ. You say lymph node. I could indicate one of these and say, name this structure. You say germinal center. I could indicate this area of the organ. where the germinal centers live, that's called the cortex. I could also point at the area where the germinal centers don't live, but I cannot ask you to name that area, which happens to be the medulla, because that's not in boldface, is it? What's in boldface is medullary cords, these lines of cells you can see here in the middle of the organ, which don't show up that well on this particular lymph node. So name the organ, lymph node. Name the structure, germinal center. Name the region, cortex. So here we have another perfectly nice lymph node. Notice the germinal center out here in the cortex of the organ. But look in the medulla. Do you see where these cells are grouped together? These are called medullary cords of the lymph node. These areas where the cells are arranged, what appear to be in lines, those are called medullary cords. So germinal centers out in the cortex, medullary cords in the medulla of a lymph node. So another lymph node that you can see right here, again, notice the fibrous capsule and these circular appearing structures out there at the edge. Let me pan around a little for you here. Beautiful ones down here. So notice these circular structures. Again, those are called germinal centers. These are characteristic of a lymph node. And then I also want you to look at these structures. In this preparation, they're stained a little darker. These walls, irregular walls inside this organ. All of these, here, beautiful one. Right there, let me move my slide around. Notice how this wall appears to be an inward projection of the capsule. These are trabeculae. An irregular network. You saw these, right? Trabeculae carnii. Remember that from the heart? Trabeculus. These are trabeculae, these walls within this organ. Now, if I go to higher magnification, so here's low power. Notice that these trabeculae are collections of fibers. I'm going to zoom in a little bit more here, in fact quite a bit more on this particular slide. Now what I want you to notice here is that I see some individual fibers within the lymph node. Let me play with the fine adjustment knob, maybe you can see them. I'll also adjust the light a little bit for you here. See these fibers right here? These are reticular fibers. You've heard of those before, way back in 223. And the primary tissue we would say that the lymph node is made of is reticular connective tissue. So individual fibers that I can see here and here in a small bundle here, making sort of a little mini trabeculus right there, these are reticular connective tissue fibers. Collectively, let me try to explain when you look at the boldface terms. All the fibers together, that's called the stroma of this organ. These cells hang around, stuck to, sidle up to, live next to these reticular fibers. Let's pan around a little more. I'll look for a spot that might be a little more descriptive for us. Beautiful right here. See these reticular fibers? So an individual fiber is a reticular fiber. The reticular fibers all together, collectively, are called the stroma. Now, I know what you're saying with an online lab. You're saying, Dwayne, an online lab has all sorts of problems. Maybe my video doesn't stream fast enough on my device. Maybe I'm getting all these buffering things. Sorry, there's not much we can do about that. But you might say, how in the heck would I know what you want if you ask this question? Well, if I had one fiber indicated, and I said, name this structure reticular fiber. If I have multiples indicated, or somewhere in my question I say all of them, then it would be the stroma. So there would have to be something in the way I indicate or in the wording of the question that would tell you, because there's only going to be one correct answer, everyone. So an individual fiber is a reticular fiber. All of them together are called the stroma. And in order for us to identify trabeculae, I cannot be at high power magnification. I would have to be at lower magnification like this. So right here, trabeculae. Trabeculae, like that. All of the fibers together are called the stroma. So this is a lymph node. Name the organ, lymph node. How will you know that? The circular structures here, here, here. The walls, trabeculae. And then on high power magnification, reticular fibers, stroma. And of course, don't forget the fibrous capsule out here. So this is a lymph node. Next, I want you to look at the very similar lymphoid nodule. And before I switch to that slide, I want you to point your eyes at these germinal centers right here. Look at those. Think about those hard, what they look like. This will be how you know a lymphoid nodule. So look at these germinal centers and think about them while I call up the slide of a lymphoid nodule. Okay, everyone, here is a perfectly great slide showing us a lymphoid nodule. Lymphoid nodule. You see that at the top on page 5, sometimes called Peyer's patches. You call them lymphoid nodules. Now, this whole slide is not a lymphoid nodule. Only this part is. Now, remember I told you to look hard at that germinal center. Focus on, think about what that looks like. A lymphoid nodule looks just like one of those germinal centers from a lymph node that has been transplanted to a digestive organ, the wall of a digestive organ. So that germinal center you see that tells you you have a lymph node, I want you to imagine that I plucked one out of a lymph node and I stuck it here in the wall of the intestine. Now, I've never taught you how to know that this is the wall of an intestine. This happens to be your large intestine. And you don't really have to know how to do that right now. We'll get that in lab number 6, trust me. But here's what I want you to think about. I had a student once a few years ago who told me that the wall of a digestive organ to them looked like flames. Flames like you might paint on the side of a car, because this particular student had a sports car with flames on it. And he said, yeah, that looks just like the flames on the side of my car. Now, I don't know if a custom paint job of flames is going to look that much like a large intestine, but I do get his point. Notice this sort of flamey look, And then I've got that germinal center planted right there under the wall. So look for this, for lack of a better term, flame-like appearance of the digestive wall, very characteristic, and a germinal center stuck right under it. This is a lymphoid nodule. And for lymphoid nodule, all I can ask you is its name. So what is this structure? Lymphoid nodule. In lecture, you can talk about why they are there, what they do for us, immunologically and lymphatically speaking, but lymphoid nodule looks just like a transplanted germinal center. Okay, here's another lymph node. Notice the fibrous capsule here. Germinal centers I can see in the cortex. But the reason I called this one up is it probably is about the best one I've seen so far that shows me what we call medullary cords, groups of cells in the middle of the organ sort of lining up. So in my lymph node. Not much in the way of good trabeculae here in this particular slide. Let's go over here. So germinal center, medullary cords, there and there, fibrous capsule, a lymph node. Okay, everyone, last but definitely not least, I show you this very nice slide of a spleen. This is the last slide in our histology section, a spleen. It's not as bean-shaped as the lymph node in a slide. In fact, the sections tend to look sort of, I think, like a tongue, sort of like an aerial view of a tongue. But what you will notice is that I do not, I'm going to repeat, not see the characteristic circular germinal centers all over around here in the cortex. I can see on this particular slide a very nice fibrous capsule, notice that, coming into the organ, forming irregular network of walls. These would all be trabeculae for this particular organ, the spleen. So I've got a capsule, trabeculae, all very obvious, I think, in this slide. And then we have this idea of red versus white pulp in the organ. Now, this one seems a little, what's the word I'm looking for here? Counterintuitive, unless I explain it first. So, unstained, this organ had areas, sections, that were much lighter in color. And those lighter tissue areas take more stain into them. So after it's been stained, the areas that used to be light in color are now darker in color because they've been stained. So when you see these darker areas here, a little bit here, this is called white pulp. Let me scroll around. So see this little darker mass right here, white pulp. Remember our staining of white blood cells. This used to be white. Now it's been stained, so it appears darker than the surrounding tissue. So the white pulp in the spleen are these areas right here, that are more darkly stained. The red pulp is everywhere else that is lighter stained. Let me look around a little bit here. Oh, perfect example right here. White pulp, everything around it is red pulp, and look at these trabeculae, everybody. Very nice, obvious trabeculae. capsule of the spleen, white pulp, white pulp, white pulp, red pulp, red pulp. So the white pulp took stain and became darker. And in most preparations, remember on the spleen, I sort of get this tongue-like appearance in many of the slides that you will see. So lymph node, lymphoid nodule, and spleen. Name the organ, spleen. Name the structure, capsule. Name the structure, trabeculae. Name the structure, white pulp. Name the structure, red pulp. Like that. You'll get the hang of it in the practice, I hope.